Hi, my name is Jeff Law. I'm a PhD student at Virginia Tech, and I'm very excited to present my work with several collaborators on identifying human interactors of SARS-CoV-2 proteins and drug targets for COVID-19 using network-based label propagation. The life cycle of SARS-CoV-2 is being studied by researchers worldwide to try to find vaccines, antivirals, and drugs to treat COVID-19. Many drugs, such as remdesivir, that interfere with different aspects of the viral life cycle are in clinical trials. Since the virus must use human proteins to replicate itself, a promising approach to identify more treatments would be to repurpose existing drugs that target the proteins or processes on which the virus relies. With that goal, a recently published affinity purification mass spectrometry study by the Krogan Lab outlined 332 proteins that directly interact with SARS-CoV-2 proteins. This dataset provides an incredible resource for some of the initial points of contact the virus has in the cell and shows some plausible drug targets. But it does have some limitations. First, the experiments were done in a kidney cell line and do not capture in vivo or conditions during infection, likely causing missed interactors or false negatives. Second, by its very nature, the method does not capture any downstream effects of the virus. Our hypothesis is that complexes or signaling pathways that are close by the initial interactors could be processes used or hijacked by the virus and could potentially present good drug targets. Thus, our goals are to first prioritize additional human proteins that may directly or indirectly interact with the virus, and second, to identify drug targets among these proteins. Our approach is to perform network propagation on a protein functional interaction network using the human interactors of the virus as starting points. We then validate our approach and analyze our predictions. First, I'll describe our approach. Network propagation can be thought of as a process where we treat the human interactors of SARS-CoV-2 as sources of heat, and then let the heat propagate through the network. This is a well-established approach and has stood the test of time in computational biology for many different applications. Here, the main idea is that the nodes that are close to many original interactors are more likely to also be affected by the virus. Thus, they'll get a higher score than nodes that are farther away in the network. Now, looking at the specifics of the method we're using, given an undirected weighted network and the starting vector of node labels, either 1 for positive examples or 0 otherwise, we compute a score between 0 and 1 for every node using the regularized Laplacian. Since this is a pretty standard approach, I'm going to skip over the explanation of the equation today, but I'll leave it here for reference. We compare this classic RL approach to several others. Gene mania and off-the-shelf gene function prediction is basically the same as RL, except it can take specific negative examples. Sync source, a method used to find human dependency vectors of HIV, is also similar, except that it fixes the scores of positive examples. We also compared a couple of standard supervised learning methods, SVM with linear kernel and logistic regression. Note that we used each node's adjacency vector as its features. And lastly, we compared it with a baseline we call local, in which a node's score is simply the weighted average of the labels of direct neighbors in the network. Now for our data sets. As I mentioned, we use the interactors of the viral proteins as starting points for network propagation. For our network, we use the string functional interaction network, which includes protein interactions, gene co-expression, and more. Note that 4 of the human interactors of the virus were not in this network. Then to identify drug targets, we use the drug bank database, which had about 6,000 drugs and 3,000 targeted proteins. To test if network propagation was a reasonable mechanism for predicting human interactors of SARS-CoV-2, we performed a leave one out propagation analysis to measure the proximity of the human interactors in the network, as well as cross-validation to gauge our prediction accuracy. In the first validation, we're trying to see if the human proteins that interact with SARS-CoV-2 are closer to each other in the network than we'd expect by chance. So what we do is we leave out one of them, treating it as an unlabeled node, perform network propagation, and then store the left out node's score as well as the scores of 1,000 other randomly selected nodes, and repeat this process for every human interactor of the virus. Examining the propagation scores on the x-axis with the y-axis showing the kernel density estimate of the distribution of the scores, you can see that the distribution for the human interactors of SARS-CoV-2 is clearly shifted to the right compared to the random proteins, indicating that the interactors are, by and large, closer to each other in the network than to random proteins, with the Kolmogorov-Smirnov test comparing its distributions showing a statistically significant difference. Now, if we perform the same evaluation for other viruses with a comparable number of interactors, namely SARS-CoV, HIV, and herpes, we see this trend is not unique to SARS-CoV-2. And this could indicate that network proximity may be a general property of human proteins that bind to a virus. Next, to compare the accuracy of our methods at predicting other human interactors of the virus, we performed five-fold cross-validation, which tests the method's ability to recover the original human viral interactors by ranking them when compared to negative examples in purple. We then compute the precision and recall for each rank, giving a curve, measure the area under this curve, and compute the early precision, or precision under recall of 0.1, which allows us to estimate accuracy for the high-confidence proteins. While there are no known negative examples of human interactors of SARS-CoV-2, they were required by a few methods and to evaluate precision. So we took the simpler approach of sampling proteins uniformly at random from the network. And to ensure that the specific negative examples didn't play too large of a role for the algorithms, we averaged their scores over 100 sets of negative examples. And for evaluation, we repeated the cross-validation process 100 times and tested positive to negative ratios of 1:1, 1:5, and 1:10. Now for the cross-validation results. Here are the distributions of early precision values across the 100 repetitions, with the dotted line showing the precision of a random predictor. We observed that RL and Gene Mania were pretty much indistinguishable and achieved the highest median precision, close to 0.9, with the others not far behind it. And the fact that the baseline method, local, does not do as well also helps confirm the benefits of propagation. Now, if we compare the area under the precision recall curve, the conclusions are essentially the same, and also for higher ratios of negative examples, which I'm not showing. These evaluations help confirm that network propagation is a promising approach for the prediction of human interactors of SARS-CoV-2. Next, to ensure we could trace the evidence for every prediction, we analyzed the propagation process to trace how much each human interactor of the virus contributed to the prediction score. I'll have to skip this today due to time, but those results are in our preprint, which I'll give a link to at the end. Now onto our main results. We analyzed the top 332 predictions of RL, and to try to remove any noise that could be present among these predictions, we designed a degree stratified sampling approach for estimating p-values and kept only nodes with a p-value less than 0.05. We then tested for enrichment of Go biological process terms among these predictions and found over 180 terms that passed the significance threshold. So then to remove most redundant terms and summarize this list, we designed a simple set cover algorithm, which greedily chooses the most significant and non-overlapping terms, and resulted in 21 terms. Here's an overview of those enriched terms and their connections to the viral proteins through the human interactors. These terms fall into 5 main groups, as you can see: organelle organization, post-translational modifications, ER stress, respiration, and transcription and translation. And interestingly, you can see that often many virus proteins work together to converge on these processes, with viral transcription and protein folding connected to 9 or more viral proteins. Then to see if there's involvement of these processes in the viral life cycle, we searched the literature for evidence for SARS-CoV-2 or other closely related viruses. And we're happy to report that for most of them, we did find literature evidence. I don't have time to get into our findings for each of these terms, but if you're interested, more information is in our preprint. Today, I'll focus on the endoplasmic reticulum, or ER stress group. ER stress is a well-documented initial response to viral infections with lethal or severe clinical outcomes, and was also shown to be activated during SARS-CoV-2 infection. One of the Go terms we prioritized, which is highly overlapping with other ER stress-related terms, is protein folding in ER. This network is showing our top predictions in green annotated to this term, with the neighboring human interactors of SARS-CoV-2 in blue, the viral proteins in red, and the drugs targeting our predictions in orange. And the green and blue edges show the edges in string between our predictions and the connections to the initial human interactors. Lastly, the highlighted proteins are ones we discuss in greater detail in our preprint. One of our top predictions and a master regulator of VR stress is heat shock protein 5, or HSPA5. Also known as GRP78, this protein has increasingly well-documented essential interactions and activities during viral infection. This includes everything from viral attachment and entry to protein production, as well as viral release. And in addition, HSPA5 has been suggested as a receptor for the SARS-CoV-2 spike protein by a recent molecular docking study. One of the surprising effects of COVID-19 is that it has been shown to cause moderate to severe blood clotting in many cases. Interestingly, we believe this network may suggest potential mechanisms by which the virus may induce blood clotting. Specifically through HSPA5, calnexin, or CanX, and calreticulin, or CalR, which are known to be chaperones for other procoagulant proteins. This raises the possibility that anti-clotting drugs may be used to treat COVID-19. In fact, acetylsalicylic acid, or aspirin, which targets HSPA5, as well as a derivative of linotidase called alteplase, which targets CanX and CalR, are already in clinical trials for their anticoagulant effects on COVID-19. In summary, we have found network propagation techniques to be well suited to pinpoint processes that may be hijacked by the virus. We provide a prioritized list of human proteins, which are supported by the literature, and drug targets, of which at least 6 are already in clinical trials. We described a promising direction to target ER stress and HSPA5 specifically. And lastly, in our preprint, we found similarly exciting results for tubulins and cilium assembly. We're currently searching for collaborations to evaluate our prioritized drugs, and if anyone is interested, we would love to hear from you. And I'm going to skip our next plans, but I'll be happy to discuss them during the questions. I want to invite you to read our preprint on archive, check out our code on GitHub. And lastly, to make the underlying interaction evidence of our predictions available to other researchers, we created an interactive network for each of our prioritized terms on the network sharing platform called GraphSpace, which you can access using that link. Finally, I'd like to give a big thanks to all the co-authors who worked on this project with me, especially my advisor, Dr. Murley. It's been a thrill working with them at such a fast pace. I want to acknowledge our funding sources, and last of all, thank you all for listening.